the first function of thyroid peroxidase is nothing but the conversion of this iodide molecule into this iodine molecule. That is nothing but the iodination of the iodide molecule. Second function of this thyroid peroxidase enzyme is nothing but the organification of this iodine molecule, that it combines this iodine molecule towards the tyrosine molecule. And the third function of this thyroid peroxidase enzyme is nothing but the coupling reaction, that it combines or it binds the two adjacent tyrosine molecule. So there are three functions of thyroid peroxidase enzyme. Hence we will draw this sequence of thyroglobin as in a simple way. Suppose this is a thyroglobin molecule, this thyroglobin molecule, we can put it like that: T3, T4, monoiodotyrosine, diiodotyrosine. So this is the one sequence of thyroglobin, and as for the example, but the each and every thyrocyte which forms the thyroid follicle secretes a number of thyroglobin molecule. This is a one particular thyroglobin molecule, and this is a simple form of that thyroglobin molecule. So this thyroglobin molecule consisting of now T3, T4, monoiodotyrosine and diiodotyrosine. So this thyroglobin molecule or the mixture of thyroglobin molecule, there are number of thyroglobin molecule. Like that: diiodotyrosine, monoiodotyrosine, T3, T4, diiodotyrosine, monoiodotyrosine. Suppose it contains T3, T4, another molecule of T3, diiodotyrosine, monoiodotyrosine, another molecule of T4, T4 molecule, T3 molecule, T3 molecule, diiodotyrosine, monoiodotyrosine, again T3. So there are mixture of thyroglobin molecule inside the lumen of thyroid follicle. This mixture is called as colloid. So the mixture of thyroglobin molecule inside the thyroid lumen is called as colloid. So this colloid once get prepared or once get formed, this apical membrane, once the mixture of colloid or mixture of thyroglobin, that is colloid, get formed, this apical membrane of thyrocyte cell again get converted into the pseudopod. So this apical membrane get converted into the pseudopod, and it takes the part of colloid inside. So this is the one part of colloid. So this part of colloid has been taken up by this pseudopod of the apical membrane of thyrocyte cell, and once this thyroglobin forms a part of this pseudopod, it further enters inside the cell. So this is the pinocytic vesicle. This pseudopod further takes the part of a colloid and enters inside taken inside the thyrocyte cell. So this pseudopod, with the help of pinocytic process, this pseudopod taken up the part of a colloid and inside the cell. So with the help of pinocytic process, this part of colloid enters into the pinocytic vesicle. So this is one thyroglobin or a part of colloid. This part of colloid which is present inside the pinocytic vesicle. So for the digestion purpose, this pinocytic vesicle has to be converted into the digestive vesicle. For the digestion purpose, this pinocytic vesicle has to be converted into the digestive vesicle. So there are numerous lysosomes present inside the cell. So this lysosomes comes and binds with this. Let's draw with another pen. go and binds to the this penocytic vesicle. So once they binds with this penocytic vesicle, they secretes their lysosomal enzymes inside. Inside there is a part of a colloid. So once this lysosomes goes and binds with this penocytic vesicle, they secretes their lysosomal enzymes inside. Once this lysosomal enzymes goes inside the penocytic vesicle, further there is a one enzyme. Suppose this is a one enzyme, that is proteolytic enzyme. So this proteolytic enzyme cuts the pieces of the T3, T4, diiodotyrosine and monoiodotyrosine. So with the help of this proteolytic enzyme, which is coming from the lysosomal system, this proteolytic enzyme further cleave the T3 molecule, T4 molecule, diiodotyrosine molecule and monoiodotyrosine molecule. So the molecules of T3, T4, diiodotyrosine and monoiodotyrosine finally get formed. So this is T3, T4, diiodotyrosine and monoiodotyrosine. So out of which T3 and T4 has to be again secreted into the blood vessel. So T3 and T4 goes and directly enters into the circulation. Finally goes into the circulation. So these enzymes, this hormones, that is T3 and T4, goes and directly enters into the circulation. The problem with this diiodotyrosine and monoiodotyrosine. So what happened next with the diiodotyrosine and monoiodotyrosine? Suppose this is the structure of monoiodotyrosine and this is the structure of diiodotyrosine. So this molecules cannot goes directly into the circulation. These molecules remains or get accumulated into the thyroid cell. So there is one enzyme, there is another enzyme. Suppose this is a enzyme. This enzyme is nothing but thyroid deiodination enzyme. So this thyroid deiodination enzyme takes the monoiodotyrosine and diiodotyrosine and it cleaves these iodine molecules from molecule of monoiodotyrosine and diiodotyrosine. So the free iodine molecules get further goes, this three free iodine molecules forms and this three free iodine molecules goes and enters into the process again and again get utilized. So this is all about the synthesis of thyroid hormones, that is T3 and T4. So in previous part we have seen the synthesis, storage and secretion of thyroid hormones. Now let's start with the new part. Usually the thyroid hormones having three important function. The first function is to regulate basal metabolic rate. To regulate the basal metabolic rate, that is nothing but the BMR, that this thyroid hormones helps in the metabolism. Second function is it helps in physical maturation, that is nothing but the physical growth. So second function is that is nothing but the physical, it helps in physical maturation. And third function, it also helps in mental maturation. So main important function of the thyroid hormones is to regulate our body's basal metabolic rate. Now let us talk about the regulation of secretion of thyroid hormones. If there is a low basal metabolic rate, if our body having low basal metabolic rate, it is first detected by hypothalamus. So hypothalamus firstly detect what? That is low basal metabolic rate. Once hypothalamus detects the low BMR, it further release one particular hormone, that is nothing but the thyrotropin releasing hormone. Once this hormone is released, this hormone further goes into the anterior lobe of pituitary gland. This is the anterior lobe of pituitary gland. So this thyrotropin releasing hormone enters into the anterior lobe of pituitary gland, and it stimulates one particular cell, that is nothing but the thyrotrop cell. This is the thyrotrop cell. This thyrotrop cell once gets stimulated, it secretes one particular hormone, that is nothing but the thyroid stimulating hormone. This is the thyroid stimulating hormone. The name itself indicates that this thyroid stimulating hormone, it stimulates what? Thyroid. That nothing but it stimulates the thyroid gland. So this thyroid stimulating hormone once enters into the blood circulation, it goes towards the thyroid gland. It goes towards the thyroid gland. It comes towards the thyroid gland and it stimulates this one particular thyroid gland. Once this thyroid stimulating hormone stimulates this gland, that is nothing but it stimulates the thyroid follicles. This is nothing but the cross section of thyroid gland. That is, it contains a number of thyroid follicles. This thyroid stimulating hormone stimulates the thyroid gland. That is nothing but it stimulates this thyroid follicle. Once this thyroid follicle gets stimulated, this thyroid follicle releases their hormone. This thyroid follicle contains a T3 and T4 hormone. So this thyroid follicle releases T3 and T4 into the circulation. Once the T3 and T4, that is thyroid hormones, enters into the circulation, their normal level get achieved, and once these hormones enters into the blood, they causes their function, that is to regulate the BMR, that is to regulate the basal metabolic rate, and its function get over. Once the function of thyroid hormone get over, it is again detected by hypothalamus. Once the function of thyroid hormone get completed, it is again detected by hypothalamus. Then hypothalamus stops the secretion of thyrotropin releasing hormone. Hence the secretion of thyroid hormones get inhibited and further cycle gets cut off. That is, this is the further cycle gets cut off. So the secretion of hormone, the cycle containing secretion of hormone called as positive feedback mechanism, the cycle in which there is a secretion of hormone takes place called as positive feedback mechanism, and the cycle in which the secretion of hormone gets stops, that is nothing but the negative feedback mechanism. Once the hypothalamus releases thyrotropin releasing hormone, that is it nothing but it releases one particular hormone, and this thyrotropin releasing hormone further leads to a release of thyroid stimulating hormone. This is nothing but the secretion of hormone, hence it is called as positive feedback mechanism. And here once the function and the normal level of T3 and T4 get completed and its function get completed, there is a inhibition of cycle, and this inhibition of cycle of thyroid hormones is nothing but the negative feedback mechanism. This is all about the secretion and regulation of thyroid hormones.